7.3 Electron Transport. So glycolysis was outside the mitochondria, produced some pyruvate. The pyruvate feeds into the preparatory reaction where we form acetyl-CoA. Acetyl-CoA feeds the citric acid cycle. Along these first three steps, we have completely oxidized glucose, produced just a little bit of ATP, but the cell has produced a number of NADH and FADH molecules. These are high energy molecules. That energy was taken from glucose, and now it is going to feed the electron transport chain, where again we make the bulk of our ATP. The electron transport system is a series of proteins. They're embedded in the inner mitochondrial membrane, and they pass electrons from one to another. So it's kind of like a bucket brigade. These electrons come from NADH and FADH2. They feed their electrons into these electron carriers, and then they're passed from one to another, kind of like a bucket brigade. And then down at the bottom of the hill is where we must have oxygen. So oxygen is the final electron acceptor in this process. If oxygen is not available, it's like putting a dam across a river. All of these reactions back upstream are going to stop if oxygen is not available. Now call timeout. This is the electron transport chain, so we are passing electrons from NADH and FADH2 to oxygen. Okay. But that NADH and FADH2 has picked up a hydrogen atom. So recall that hydrogen consists of an electron and a proton. What's occurring here is the electrons are being passed to oxygen, so we need to account for the protons. So here is the really neat trick that occurs. Those protons are going to be pumped across the inner mitochondrial membrane, and so they are between the mitochondrial membranes. This is called the intermembrane space. So the energy from these electrons, as they are being passed from one carrier molecule to another, is used to pump hydrogens into this intermembrane space. And it creates a very high concentration of protons. It creates a concentration gradient, and that concentration gradient contains energy. Those protons are going to try and diffuse down their concentration gradient. They will try to go from high concentration to low concentration. However, they are charged, and recall, charged particles cannot diffuse through the lipid bilayer of the membrane. So the only way that these protons can diffuse down their concentration gradient is to pass through a molecule here in the bottom of the diagram that is called Atp Synthase. As these protons pass through Atp Synthase, we are going to add a phosphate to adenosine diphosphate to form adenosine triphosphate. So the bulk of our ATP is made right here in this step. Those protons are pumped into the intermembrane space, then they diffuse back through the ATP synthase complex to get back inside the matrix, and these protons along with the electrons are then added to oxygen to form water. Here's a diagram that shows the energy yield from glucose, kind of summarizes the steps of the process that we've just looked at, and also summarizes where the ATP is produced, the NADH, the FADH2. So beginning in the upper left-hand corner, the first step was glycolysis. So glucose is split into two molecules of pyruvate, so the cell can net two ATPs from glycolysis. there is also some NADH that is produced. That NADH can then be transported into the mitochondria to feed into the electron transport chain. Some cells have to invest a little bit of ATP to get that NADH across the mitochondrial membrane. So there is some variability in the ATP yield due to this shuttle in the mitochondrial membrane. But the pyruvate that results from glycolysis produces additional NADH along with the acetyl-CoA. The acetyl-CoA fed into the citric acid cycle, as we saw earlier in the lesson. And each of these steps is producing NADH, FADH2, or ATP. Ultimately, those electron carriers, NADH and FADH2, feed into the electron transport chain, as we have just seen. So the net yield of ATP from cellular respiration is either 36 or 38 molecules of ATP. Again, the variability is due to the shuttling that has to occur to get the NADH into the mitochondria. 38 ATP molecules, this represents about 40% of the free energy from glucose, so about 40% efficient. It doesn't sound terribly efficient. However, if we look at something like burning gasoline in the engine of a car, that's only 20 or 25% efficient. Recall all of these energy conversions, we discussed this back in lesson 6, all energy conversions are going to be inefficient. And so we lose some useful energy as either heat or entropy. And so again, we end up with about a 40% efficiency in the oxidation of glucose.